Creating a visualization is not as simple as dragging and dropping in a structure to render. The colors, pose, style, and theme will vary based on the story you are trying to convey and the audience to which you are conveying. In this third installment in our PyMOL tutorial series, you will learn techniques necessary to make visualizations as aesthetically pleasing as possible, while also thinking about the accessibility and the many options in PyMOL. This tutorial is also available in a written format in the description down below. In this tutorial, we will first cover positioning and representation types, then coloring, and lastly, we will finish up with a catalog of representation techniques that may be useful during your use of PyMOL. Let's begin. How a structure is represented in pose can drastically change the quality and message of a visualization. This first section will cover some good and bad examples of positioning and representation being used. Some of the steps performed here will be covered in less detail than in previous tutorials. If you are struggling or need help, click the link in the description below to go to our playlist for our tutorials. As done in previous tutorials, we first need to fetch the structure we're using. Click File, Get PDB, and type in the 4-letter code 5ZHP. This is a structure of an acetylcholine receptor bound to a selective agonist. Now, where it says chain name, click this button and select A. Now click Download. Now click this middle bar to drag it up to the top and make it a little larger. Now we have to enter in our initial settings. Hit Display, Background, White to set the background to white. Now click Display, Show Valences to turn valence off, and then type in set cartoon underscore SIDE side and hit tab. So cartoon side chain helper, and then type 1 to turn cartoon side chain helper on. And lastly, remove the hydrogens by typing in remove hydro and then enter. We're now going to color the receptor and ligand some temporary colors just to aid in the visibility while we're testing a few poses. Next to 5ZHPA, click C for color, blues, slate. Now, right click and drag to zoom in on the ligand, which is the bottom stick portion here, not this top portion. It's the larger one down here in the bottom. Once you clicked it, next to Celly, hit C for color, oranges, orange. Now next to 5ZHP again, hit the C for color, by element, and then the first option, which is HNOS with no C. Once again, this is just temporary while we're testing some poses. When posing your molecule, it may be challenging to know where to start or where to look for information about the molecule. One of the easiest places is just to go to the website rcsb.org, which houses the structure, and then look up the 4-letter code associated with it. So 5ZHP. Next to the molecule, you will see something that says literature. This houses the paper associated with the PDB file we're working with. If you click Search on PubMed, it will link you to the literature associated with the PDB. Clicking PMC, which stands for PubMed Central, will open up the paper for this molecule. If you scroll down, you will see a figure associated with the structure, and it actually has some information about how to pose the molecule itself. You can see the ligand there, you can see the intracellular, the extracellular portions of the molecule. I highly advise you take notes on what you see in this paper and read through it, as the information inside will help aid you in what you want to highlight and not highlight with the structure and what you may want to convey to your audience. Now back to our PyMOL session. You can see what the paper was talking about when it mentioned an extracellular and intracellular portion with the ligand being right here. What you want to do is think in your head about the story you want to convey to the audience and how to best pose the molecule to portray that story to your audience and push that narrative. The information gleaned from the literature will vary depending upon the individual reading it. However, for the purposes of this tutorial, there's three main goals we want to accomplish with the story we are telling. The first of which is ligand visibility. The location of the ligand in the receptor has to be as visible as possible, and even though the ligand is quite a bit smaller than the receptor, we want the structure of the ligand to be as visible as possible as well. The second is receptor visibility. This means we want as much of the receptor structure in frame, and we want the orientation of the pose to show as much of the secondary structure as possible. Lastly, directionality. Directionality is the use of specific orientations, i.e. up, down, left, right, to convey a specific message to the audience. For example, in this case we have an extracellular and intracellular portion of the molecule. The human mind will make assumptions based on the orientation of the pose we choose. So, if we have the top part of the molecule as the extracellular portion, the human mind will assume that that part is out. And if we have the intracellular portion facing downward, then the human mind will assume that is in. Here are a few examples of some good and bad poses. Here is an example of a bad pose. Although the ligand is very easy to see, the receptor looks clumped due to bad positioning. In addition, directionality is not being used to show the intracellular from the extracellular portions. Here is yet another example of bad posing. Although the receptor is in pretty good view, and you can see two portions of it, the directionality is not clear here. You can see one portion and another, but it's not clear which one's intracellular and extracellular without adding things like labeling. In addition, the ligand is obscured by the receptor, making it very hard to see any of its structure. Here is an example of a suitable pose for the structure. First, the ligand is in clear view in the receptor. Not only can you see its structure, but you can see where it fits in the receptor. Second, the receptor's secondary structure is very visible. You can see the shapes and the distinguishing parts between the top and bottom. In addition, the directionality is being utilized here to emphasize that the extracellular portion is up and the intracellular portion is down, making it more apparent which is which. When you find a pose you like, click Get View. This prints out coordinates on the command line that you can cut and paste into the command entry later to retrieve your camera's position. In addition, it also copies it to your clipboard to use immediately. By hitting Ctrl V, you can paste and reset the camera even if you've altered it. Here you will notice the pose chosen is pretty similar to that of the literature. This will not always be the case, however in this instance it's a pretty useful and descriptive pose. Now I'm going to take a step back from the PyMOL session and go over representation types a little bit. There are various options in PyMOL for representing structures. There are appropriate and inappropriate times for every representation type. Appropriate and thoughtful usage ensures the structure looks as professional as possible. You are welcome to follow along by tinkering with your PyMOL session, however you are also welcome to just observe some good and poor usage examples following this. When you hear sticks, you should think detail. This is because sticks represent each individual atom instead of something like cartoon, which shows an overarching shape for everything. It is because of this that they are specifically useful for emphasizing small features of a structure, and for this reason, they are the default representation type for smaller molecules in the structure that aren't amino acids or nucleic acids. Here's an example of good usage of sticks. Here the sticks are being used to represent the ligand since it is a small molecule, and also being used to emphasize the residue it is interacting with. Once again, sticks are being used as detail. We would normally only see this portion of the protein as a ribbon rather than the sticks, but now we can see the detailed protrusions. In addition, we can also see the individual atom types because sticks have representation for every atom in the structure rather than the overarching curls of a ribbon or a beta sheet. Here is an example of poor usage of sticks. As you can see, the sticks are being used to represent the entire structure. Since sticks are designated for more detail-oriented parts of the structure, it doesn't really make much sense to use it for the entire thing, as all of the different sticks tend to clump together and it's hard to distinguish any real structure here. Lines are similar to sticks in that they are very detailed-oriented. However, they are rarely useful as they are so thin that they're hard to see and tend to look messy. However, they can be useful when used in conjunction with sticks. This first example is a good usage of the lines. Here, the thinness of the lines are being used in contrast with the thickness of the sticks. This emphasizes the fluorine atom that's protruding out of the ligand. Similar to sticks, using lines to represent the entire structure or even large parts of the structure is not advised, as it's very hard to see any of the detail. It gets lost in the mess of all the lines. Cartoon is the default representation type for nucleic acids and amino acids. It can't be used to show anything other than these two residue types, but instead of cartoon, ribbon is an option. This isn't very useful for making informative structures, as beta sheets and alpha helices aren't explicitly shown when using ribbon, but this can be useful when you're trying to change up the style of a visualization. For most applications, however, it is recommended that you use cartoon. Here's an example of using ribbon. As you can see, you can't really clearly distinguish a whole bunch of the secondary structure. I mean, these are obviously alpha helices, but in a more complex protein, it might be a little bit more challenging to distinguish them. However, it does produce a different visual style that might be preferred depending upon the application. Spheres are the default representation for most single atoms in a structure and are sometimes used for part of small molecules such as ligands. Spheres are useful for showing the N and C termini of a protein, which I will demonstrate further in the tutorial, for single atoms in the structure, and sometimes for emphasizing certain aspects of the structure, like space of occupancy. It is not recommended to use spheres to represent more than a few atoms in a structure. On screen is an example of good usage of spheres. As you can see, the sphere is being used to highlight the fluorine atom, which, similar to the lines usage above, emphasizes that portion of the ligand. Here's a poor usage of spheres. Just like lines and sticks, it's being used to represent the entire structure, which isn't very useful because you don't get too much detail here, and you just see a bunch of spheres instead of actual features such as alpha helices and beta sheets. Now that we've reviewed some best practices and common mistakes, it is time to implement them into the molecule we fetched earlier, 5-ZHP. So let's return to the session. First, we need to color the ligand that we showed earlier back to slate. Do this by zooming in, left-clicking on the ligand. Now next to Celi, hit the C for color, blues, slate, and now once again hit the C, by element, HNOS. Now we must hide all of the small molecules except for the main selective agonist, 9EC. This can be done a couple different ways, but the easiest to do it is by using the sequence bar. Display the sequence bar by hitting Display, Sequence, and now go to 3-letter mode by hitting Display, Sequence Mode, Residue Names. Using the scroll bar, navigate to the very rightmost part of the sequence. Here you should see PG4, MAL, and 9EC. If you read the rcsb.org website, you should see that 9EC is the main selective agonist, and MAL and PG4 are just other molecules occupying that space. Click PG4 and MAL, and then next to Celi, hit H for hide, everything. Now, if you look at the molecule, you can see only the main selective agonist, which is 9EC, is visible. Now we will apply one of the representation techniques we covered in one of the previous sections in order to represent this ligand in a better way. So using the right click, you can zoom in to where the ligand is, and now make sure your mouse mode is set to atoms. Click 9EC on the sequence bar and hit S for show, lines. Now hit H and click sticks. Now deselect the selection by clicking the Celi box, and then carefully click the fluorine atom, making sure your mouse mode is set to atom once again, and the carbon right next to it. The reason we click not only the fluorine but also the carbon atom adjacent to it is because we're going to represent this as a stick, and in order to represent sticks you must have at least 2 atoms. Now next to Celi, hit S for show, sticks. As you can now see, we have the ligand represented by lines and then the fluorine represented by sticks to emphasize its importance, as we'll get into here in a second. Now we will highlight the residue interacting with fluorine by showing it as sticks. First, navigate to residue 225 by using the scroll bar. It should be the one directly left of serine, a leucine. Clicking that will make it our selection. Now if we zoom out by right-clicking, we can see the leucine right there. Now next to Celi, hit S for show, and then sticks. Now, normally you'd read through paper and see this interaction, but for the purpose of this tutorial, just choose the leucine. And keep in mind, the types of residues interacting with your ligand will vary depending on the structure. We've correctly used cartoon by showing the protein. We've also used sticks and lines to represent the ligand. Now we'll use spheres to represent the termini of the protein. There are two termini in a single chain of a protein, the C-terminus and the N-terminus. The C-terminus is where the sequence terminates or ends in a carbon atom on the backbone. This will be located on the left side of the sequence. The N-terminus is the side of the protein that terminates or ends with a nitrogen on the backbone, and it is located on the rightmost part of the sequence. First, we will show the C-terminus. Do this by clicking threonine 63, which can be located by scrolling leftmost on the sequence and counting up from 61 to 62 to 63, threonine. After you click it, it will make a new selection called Celi. Next to Celi, hit the S button for show, main chain, and spheres. The reason we do not hit S and then spheres is because it will show every atom in the residue as a sphere rather than just the backbone atom, which is what we want. Now, next to Celi, hit C for color, blues, and density. Now, navigate to the very rightmost part of the sequence by using the scroll bar and click on tyrosine 118. And click on tyrosine 1118. Now next to Celi, ensuring that you still don't have the C-terminus selected, click S for show, main chain, and spheres. Now click the C next to Celi, reds, and fire brick. Based on the standards used in Bevan Brown Laboratory, we suggest that you always color the C-terminus density blue and the N-terminus fire brick red. Now on to colors. Colors are important in visuals as they can be utilized to portray meaning. The colors you choose and where you use them will convey a message to the reader about the objects you are coloring. Before coloring, it is important to state your purpose. It can often be challenging to know where to start when picking colors for the visualization. So, ask yourself the message you are trying to convey or the purpose of your visualization. Here are a few examples of colors being used to portray different meanings with the same protein we're working on. This first example emphasizes the importance of contrast when attempting to convey a message. Here you have a light top color for the extracellular portion, and then you have a darker, more vivid blue at the bottom. This contrast highlights the difference between the top extracellular and bottom intracellular portions. In this instance, contrast is being used between the ligand and the receptor. Notice a lack of contrast is being used between the intracellular and extracellular portions of the protein. What this does is puts the emphasis on the ligand binding to the receptor rather than the two portions of the protein. In the first example, one could argue the contrast was being used to highlight the receptor. In the second example, one could argue that the contrast is being used to highlight the ligand. In this instance, we're using a lack of contrast, meaning the colors we've chosen are analogous. Although you can still clearly distinguish the ligand, the colors are that of a very similar shade, although different levels of brightness, so that neither the receptor or the ligand are highlighted. This is very useful if you're trying to emphasize the overall structure of a protein with the ligand bound in it, not necessarily having an emphasis on either the ligand or the receptor itself. Now back to our PyMOL session we're working with. In your head, think of what a purpose you might want to portray in this receptor structure might be. Before we go into coloring, however, I want to cover choosing palettes. In the description down below is a link to Adobe Color. This is a website that aids you in choosing color palettes. Although you can choose colors on your own, when starting out I recommend using a website such as Adobe Color, as it helps you get a grasp for how to use colors appropriately. Adobe Color works by dragging this center color around and assigning colors next to it based on the palette type you've selected. I'm going to choose a color and then I'll go through what each palette type does and what each one is useful for. Let's choose this center blue color right here. The first color type is analogous. Analogous consists of several tones close to each other on the color wheel. Choosing colors off of analogous palette can be useful if you're looking for colors without a lot of contrast, but still different enough to stand out. Next is monochromatic. This palette type consists of different shades of the same color. This is useful when little to no contrast is needed, similar to analogous, such as highlighting key residue sticks protruding out of the receptor shown as cartoon. If you take a look at monochromatic versus analogous, they both have very little contrast. The difference is that analogous will choose different colors around the circumference of the color wheel, but monochromatic will only vary the brightness of the main color you choose. The triad color palette has high contrast as it selects colors on a triangle basis around the color wheel. This palette type is useful if you want to make objects pop. However, the colors may not always suit each other aesthetically. However, this will vary depending on the palette. The complementary palette chooses a color opposite on the wheel of the main selection you chose earlier. This is useful as it adds a lot of contrast, and often, due to the way that colors work, it looks very aesthetically pleasing together. Once again, this is useful to make things pop similar to triad. The difference here is that the colors are probably going to look better on a complementary palette versus a triad palette. The compound palette is similar to complementary, as you'll notice there's colors on the opposite side of the wheel. However, it adds extra variation to the colors it selects. This is one of the most useful palettes, as it provides plenty of variation in the colors to use, while still having enough contrast, but still looking aesthetic. This is also especially useful for visualizations that have lots of different objects, since you have a lot of variation among the color wheel, but you still want a little bit of togetherness by having the colors look aesthetically pleasing close to one another. Shades is very similar to monochromatic, except you'll notice that monochromatic changes the saturation more than shades does. They're both used in similar applications as well. They're more useful to interchangeably be used if you want to see what a different color palette would look like. It's also worth noting that you can adjust the darkness or brightness of each color on the color palette. However, the angle between each around the circumference of the color wheel will remain the same. In addition to choosing your own palette, you can always hit the Explore tab on Adobe Color. This will provide you with an array of palettes to your liking. Here are all the color palettes lined up. See some you like? Some you don't? Good. That's because colors are very subjective. You can use color science to maximize the likability of your palette and make sure it looks as aesthetically pleasing as possible. But keep in mind, individuals have different preferences. If you have a different preference, great. That's the great thing about visualizations. You can customize them and put your own personal touch on them. Now that we've gone over the process of choosing a purpose for your project, as well as designing a color palette, let's apply both. First, the purpose. Going back to the literature, we know a couple things about the protein that we want to show. Not only do we need contrast between the extracellular and intracellular portions of the protein, but we'll also need some contrast between the ligand and the receptor itself so it stands out. And lastly, we're going to want a slight amount of darkness for this residue here, leucine 225, in order for it to stand out from the receptor. If it's the same color as the receptor, sometimes the sticks representing that key residue can get lost. Based on our purpose, the best color palette type to choose is compound. If you look at our purpose, we need enough contrast to distinguish different types of colors and different objects. However, it's still useful to have different shades of those contrasting colors in order to add some variation. I'll go further into depth as to why I chose these colors as we implement them in the visualization. The colors you choose may vary, which is fine. I just happen to like this color palette. going back to the fact that colors are subjective. Now we will apply our color palette to suit our purpose. First, we will color the protein to differentiate the extracellular and intracellular portions. We can leave the extracellular portion the slate color as it's very close to the first color in our palette. We will color the bottom intracellular portion the third color in the palette, as the light blue and the darker shade of blue will be different enough to separate the two halves of the protein. In order to color the intracellular portion, make sure your mouse selection mode is set to atoms. Now, click Mouse, Emulate Maestro. What this will do is change your mouse options to allow you to click and drag a box to make selections. Now, click and drag a box across the bottom portion of the protein. Ensure you only have the intracellular portion of the protein selected. Now, click 3-button Maestro at the bottom right of the screen to change the 3-button mode to viewing. Lastly, we will click the red sphere that's representing the N-terminus so that we don't accidentally color the N-terminus something other than firebrick red. Now next to Celly, hit the C for color, blues, and click light blue. You'll notice that this color isn't quite right. It doesn't line up with the palette selection we want. In order to make this closer to the color on the palette, we're going to hit Setting, Colors, and you can actually type in LIGH to go to light blue. And from there, we're going to adjust the red, green, and blue numbers in order to produce a color closer to what we want. You can either use the up-down arrows, drag, or enter in a value for the red, green, and blue. You're welcome to discover which color you like on your own. However, for now, set it to these defaults. When you're done, hit Apply. Now you'll notice the color of light blue is much closer to the color of the palette. Now we will color the residue 225, which is a leucine, a darker color of slate so that it stands out from the receptor. Making sure you're on 3-button viewing instead of Maestro, as it makes zooming in and out a different button. Right click and drag in order to zoom in to leucine 225. Now you can either hit mouse selection mode and change it back to residues and click the leucine, or you can go to leucine on the sequence and select it that way. Once you have the leucine and only the leucine selected, hit C for color, blues, and deep blue. Now you can see it is color number 2 on the palette, which makes it stand out from color number 1 just enough to make it pop without adding so much contrast that it looks like a different entity altogether. Now we will use the 5th color in the palette to color the ligand. Similar to selecting the leucine, we can either click it directly on the sequence, it's called 9EC on the sequence, or we can hit mouse selection mode, make sure it's selected to residues, and then click the ligand directly. Now next to Celly, hit C for color, reds, and brown. You can see this colors the entire ligand brown. However, we want to make sure we still can distinguish the different atom types. So now next to Celly, hit C, by element, and the first one, HNOS. And that's actually all the coloring we need to do. You'll notice we didn't use the 4th color in the palette, and that's okay. Sometimes you just don't need all the colors. However, if you take a look at the visualization, you can see we've used colors pretty effectively here. We've used contrast between the color number 3 and number 1 to highlight the difference between the intracellular and extracellular portions of the receptor. We've used very little contrast to highlight the key residue leucine 225 interacting with the ligand. This darker shade of blue that's slightly darker than the slate is important because it makes it stand out without making it a completely different color, so it still looks like it's part of the extracellular portion of the protein. And lastly, we've colored the ligand a dark color that's contrasting from the extracellular portion it's near. This helps to make the ligand pop and reinforce that this is a different structure within the entire receptor. And now we've got two different options on how we want to make a render for this. We can either zoom in and make the focus of the render be the interaction of the ligand with the key residue leucine 225. Now, of course, normally if this was a real visualization I was doing, I would make a measurement and label. However, since we've previously done that in tutorial number 2 with labeling and measuring, we won't cover that here. Alternatively, I could pose the picture this way, so I have the bottom and the top portion being the main focus of the story I'm trying to tell with this visualization. As always, you can hit the Draw Array button at the top to ray trace. In this next section, I'm going to go over a couple representation techniques that might be useful depending on the visualization you're performing. This will serve as a sort of catalog that you can go through and look back at to reference later in case you want to implement these settings into your session. In the description down below will be a timestamp for each representation technique covered in this catalog. The first thing I want to go over is cartoon texture. By clicking on Setting, Rendering, and then Texture, you can see a couple options here. What these options do won't necessarily be very apparent as soon as you click it. You'll need to ray trace to see. What I'll do is I'll ray trace on each of these settings and show you what each of them looks like with this identical pose. Now I want to go over shadow modes. By hitting Setting, Rendering, and Shadows, you can select from a couple different shadow options. Here are what those options look like. Next is turning on Cartoon Fancy Helices. By hitting Setting, Cartoon, and Fancy Helices, you can see the cartoons now have a ridged outer edge to them. This makes them look a little higher quality for publications. Now I want to show off cartoon highlight color. I think this looks better with Fancy Helices turned on. However, you can always turn on cartoon highlight color without Fancy Helices turned on. What this will do is add a secondary color to the back of each of the cartoon sheets and helices. Let me show you. Hitting Setting, Cartoon, and Highlight Color will turn the highlight color to on. You can change the color by hitting Set Cartoon _ highlight color and then hit Enter. I think the white does a good job at making it pop a little bit. If you don't want to enter anything in the command line, you can always just hit Setting, Edit All, and then search Cartoon Highlight Color and enter any value you want to. Make sure you hit Enter when you're done. To turn it off, simply just hit Setting, Cartoon, and then Highlight Color once again. The next thing I want to cover is ray trace modes. Ray trace modes can be adjusted to create different styles for rendering. They can be changed by using the command line and typing set ray underscore trace mode, and then typing in a value from 0 to 3. You can also adjust the ray trace mode by going to Setting, Edit All, typing in ray trace mode in the search bar, and then entering in a value from 0 to 3 as well. Here's what each of those settings look like. It is worth noting that 0 is the default. Default appearances for representation types such as sticks, spheres, and lines are not always sufficient for a visualization. The sizes of them can be adjusted using the command line or the Edit All menu. First, I'll show you how to adjust spheres. In the command line, you can type set sphere underscore scale and a comma and adjust the value to what you want. Alternatively, you can go to Setting, Edit All, and look up sphere underscore scale and adjust the value that way. Here's what a couple different options with sphere scale changed look like. To adjust the size of sticks, simply go to the command line and type in set stick underscore radius and adjust your value. The default is 0.25. Similarly to spheres, you can also go to Setting, Edit All, and look up stick underscore radius and adjust the value that way. Here's what a couple options look like with stick radius altered. Lines are very similar to sticks. They're basically identical. The only difference is that their radius is set to a smaller default of 0, which results in them looking as small as possible in PyMOL. To adjust them, simply use the same options as the sphere or the stick by typing in set line underscore radius, then comma, and your value. In addition, you can also edit this in the Setting Edit All menu and adjusting your value here. Here's what a couple options look like with the line radius changed. Another technique worth mentioning here is that you can select parts of the object that you are representing and selectively make parts larger. So if I change my mouse mode to selection mode atoms, and then click the fluorine atom and then the carbon attached to it like we did previously in the tutorial, I can actually type in set stick underscore radius like I would, and let's make it 0.45, but I can add an extra argument celly so it'll only make larger the stick radius of the atoms I've selected. Now, something worth mentioning here is you can't do this within the Edit All menu that I was showing previous. You have to do this in the command line. The last thing I want to mention is coloring using commands. So if you see here, I have this leucine 225 and I have it colored a different color than the cartoon backbone it's on. Now there's nothing necessarily wrong with this image here, but you can see that the color seeps into the backbone and might not be as aesthetically pleasing as what we're looking for. To fix this, we can actually change the default cartoon color. So in this case, we want all of our cartoon to look slate, and so we can type in set cartoon underscore color, comma, slate. And so now it will overwrite any other coloring we have in order to make sure that all of our cartoon is represented by the slate color. Keep in mind that now if I try to click parts of the cartoon and color it manually, it won't work. It will always set it to slate. If I want to change this, I can just hit set cartoon color, comma, default, and it'll change it back. The interesting thing about this is this can be done for plenty of things. Sphere color, line color, dash color. So for instance, if you're going through making every single measurement a specific color in your PyMOL session that you have represented by dashes, instead you can just set the dash color to globally change that color within your PyMOL session. And this actually concludes our tutorial today. If you have any questions, post them in the comments down below and we'll get back to them as soon as possible. Make sure to check out our other videos or look back at other tutorials if you have any questions. Thank you.